My name is Pascal Derkinderen. I am from Nantes University Hospital. In my presentation, I will be talking about the pathology of the enteric nervous system in Parkinson's disease. As a beginning, I will start with a very short reminder on the enteric nervous system. The enteric nervous system is a mesh-like neuronal network organized into two main plexus, the myenteric and the submucosal plexus, composed of neurons and enteric glial cells, which extends along the entire length of the GI tract from the lower esophagus to the rectum. The neurons of the myenteric plexus control gut motility, while those in the submucosal plexus are primarily involved in the regulation of secretion and blood flow. This neuronal regulation of GI function is mediated by specific neuromediators synthesized by functionally defined enteric neurons. Among the most common neurotransmitters in the enteric nervous system, nitric oxide is often found in inhibitory muscle motor neurons, while acetylcholine is found in excitatory motor neurons. This explains why, apart from their morphology and their functional properties, enteric neurons are commonly classified according to their neurochemical coding. One of the main characteristics of the enteric nervous system is that it is readily accessible in living subjects. In sharp contrast to the CNS, the ENS can be easily biopsied in living people. Let's say, if you want to analyze the ENS, and especially the submucosal plexus properly, there are a few tips and tricks that you need to follow. The critical issue is that you shouldn't process your biopsy in the usual way. Instead of embedding your biopsy in paraffin, you will have to dissect it under a stereo microscope. The biopsy is stretched, pinned flat, then the epithelium is peeled off, that's number 5, and then you get access to the whole submucosal plexus as a number 6. Then you can stain your submucosal plexus with a neuronal marker, as shown in 7, and you get a very nice neuronal network with ganglion and interganglionic fiber. So, what is our current knowledge on the ENS in Parkinson's disease? Using phosphoalpha-synuclein immunostaining, it has been shown that the presence of phosphoalpha-synuclein histopathology, that we will call PASH for the rest of the talk, that PASH in the gut was more frequent and more widespread than initially realized. PASH is found in large proportion of PD patients with rostrocaudal distributions, the lesions being more dense and more severe in the upper GI tract than in the colon and in the rectum. The aspect of the staining in the myenteric plexus is quite similar to Lewy bodies, while the staining in the submucosal plexus is more reminiscent of Lewy neurites. It has also been shown that alpha-synuclein pathology could be detected in GI biopsies using either the classical immunohistochemical approach or new techniques for the amplification of pathological proteins, such as PMCA. That said, there are still many pending and unanswered questions about the ENS in PD. For example, we still don't know if PASH is present in only in a phenotypically distinct subset of enteric neurons. Besides the well-known cholinergic and nitrogen neurons, there is also a small proportion of dopaminergic neurons, and it still remains to be determined if these neurons are affected or not. One logical associated question is the possible role of PASH in the development of GI symptoms, which are so frequently observed in patients with Parkinson's disease. We still don't know if the presence of PSH is responsible for neuronal dysfunction that would in turn lead to GI motility disorders. Another aspect, it is well known that Lewy body and alpha-synuclein purified from postmortem PD brains promote alpha-synuclein pathology and neurodegeneration when intracerebrally inoculated. However, the pathogenic capacity of enteric alpha-synuclein aggregates remains unknown. In the same way, a comprehensive analysis of pathological alpha-synuclein in the GI tract has not been performed. There is one study which analyzed in detail the alpha-synuclein and the post-translational modification of alpha-synuclein in the appendix of the gut, of the human gut, but no analysis has been performed in the adjacent GI tract regions. So there was a critical need for these experiments. And finally, as you know, this is maybe the might be called the $1 million question. Does Parkinson's disease start in the gut? Does Parkinson's disease start in the ENS? As you are aware, there has been a plethora of studies on the gut as a possible starting site for Parkinson's disease. At first glance, most of the existing studies are quite convincing and support either a gut to brain or brain to gut transmission of Parkinson's disease pathology with a critical role for the vagus nerve. It should be, however, borne in mind that most of these experiments have been carried out after injecting large amounts of alpha-synuclein in the microgram range into the GI tract of either rodents or non-human primates, while the measured concentration of alpha-synuclein in the interstitial fluid of the brain is only within 1 to 4 or 5 nanogram per ml. It is therefore quite hard to believe by any stretch of imagination that these models could mimic what happens in Parkinson's disease patients. So is there any alternative explanation? I don't know, but maybe there might be one. The hypothesized ascending progression theory of Parkinson's disease might only, in fact, represent a simultaneous and concomitant susceptibility to disease mechanisms of the neurons of the CNS and the CNS. So let's say that in brief, the two nervous systems could degenerate in parallel without spreading of pathology. So what are the key points? The GI tract, there is no doubt that the GI tract is an interesting organ in Parkinson's disease. It might play a role in the development of the disease or as a disease modulator. And as you know, it might be an interesting source of markers for disease diagnosis and disease progression. We know that it is affected by the pathological process in a large proportion of subjects with Parkinson's disease. However, there are still many pending questions on its role, not only in the development of the disease, but also in the pathophysiology of the GI disorders so frequently observed in PD patients. And the last point, I think, is a critical one. I think we should be cautious and not draw hasty conclusions on the fact that the GI tract is the initiation site, the starting site in Parkinson's disease.